to start actually just a little bit ahead of on time, so we save as much time as possible for Dr. Meyers to speak to us. We're pleased that all of you can be here today. We're especially pleased that Dr. Craig Meyers is here with us today. I'm sorely tempted to say a lot more, but I'm not going to do the introduction until we have the introduction. So, we first would like to ask Kyle Tressner, who is our representative on the Alumni Council, to give an invocation for us. Unfortunately, my wife's out of town today, and she was asking me on the phone this morning about Dr. Meyers. I said, well, he graduated in 1982 here, and she said, oh, he's young. I said, no, he's just younger than us. Anyway, if I could ask Dr. Meyers to come up here for a minute. We really appreciate you coming, and we'd like you to take this home with you as a small token of our thanks for you coming today. And we're proud of you and the example you are for all of us and for the students especially. Thank you. Rich Robinson from the Department of Micro and Molecular Biology will now formally introduce Dr. Meyer, and then from there we'll go straight into his presentation.
So it's my honor to introduce Dr. Craig Meyers. He's the recipient of our college Alumni Achievement Award and our speaker today. Dr. Meyers received his bachelor's and master's degree in microbiology from BYU, and then earned a PhD from UCLA, followed by postdoctoral work at the University of Chicago. He joined the Penn State College of Medicine in the Department of Microbiology and Immunology in 1993, then rose through the ranks to full professor in 2002. During the past 20 years, Dr. Meyers has achieved national and international recognition for his visionary work on human papillomaviruses, now known to be major causes of cervical cancer and other types of malignancies. In the early 90s, using an innovative organotypic culture system to grow fully differentiated human epithelia, Dr. Meyers was the first individual to grow infectious HPV in vitro. This resulted in a seminal 1992 publication in Science, and this technique remains today the only method used to grow native HPV in the laboratory. His discoveries have advanced our studies in all facets of HPV biology, including structure, replication, oncogenesis, disinfection, and immune protection. And Dr. Meyers has sat on many NCI and NIH study sections as a reviewer for numerous prestigious journals, has received over 32 grants, and currently manages several $1 million in funding. And he remains incredibly active. In the past 2 years, he's authored over 15 publications. And he and his wife, Tina, have raised 4 boys, with the last one currently a student here at BYU, and 3 of them are with us today. One's in the Air Force and couldn't make it, but that was, I think, a nice surprise for Craig to have his boys here. I've known Craig for about 30 years. I can't believe it's that long. And it's been my honor to collaborate with him on a few projects over the years. And please join me in warmly welcoming Craig back to BYU.
Well, as my wife knows, I really don't need a microphone usually. Just thank everybody for this wonderful honor. The students here, when I was here, I never thought anything like this would ever happen. In fact, up to a few months ago, I never thought anything like this would ever happen. So it's been quite an honor and a pleasure to be here. And I understand there's some down the hill in another room watching, so welcome, thank you for attending. I'm going to really try my best to be on time. I'm usually pretty good on that, but so we'll just start. about 30 years ago, I graduated from BYU with my bachelor's degree. At that time, we only had one child. 30 years later, we haven't really changed too much, I would say. I would also like to mention that this is the first time since my parents' funeral that all my brothers and sisters have been together in the same room. So this is indeed an honor to have this opportunity for all of them to be here, because I usually, in my home ward, I talk to the primary a lot, and I tell them I not only love my brothers and sisters, I like them. And I think liking sometimes is more difficult than loving. And liking is important, and I look forward to the opportunity of always being with them. And if you're wondering who I am up here, I'm the good-looking one. So we like to call our lab warts are us. We work with a lot of wart viruses, genital anal warts, and so even giraffes like Jeffrey of Toys R Us, they can get warts too. This is kind of the required slide you always need to show that the reasons why it's important to study human papillomavirus. Cervical cancer is the second leading cause of cancer and cancer death in women in the world. And in fact, in some third world countries, it's the leading cause of cancer death in that country. And when only half the population can get the disease, that's an awful lot of women that are suffering and dying from this disease. But you can also see here that several other cancers are associated with HPV. And in fact, oropharyngeal cancer is now on the increase. The typical cancer of the throat is going down due to alcohol and tobacco, and HPV-associated cancer of the throat is going up. In fact, the only two cancers that have increased in the last few years in the United States are oral HPV and anal HPV cancers. HPV has a very interesting life cycle. It requires fully differentiating epithelium, whether it's cutaneous or mucosal epithelium. There are types that can replicate it each. And so in the 1930s, when papillomavirus was first described, it was the first DNA tumor virus ever described. Nobody from that time point until 1992 could actually grow this virus in the laboratory. So if you're working on an infectious agent and you're trying to study it, it becomes very difficult if you can't grow it in a culture dish. And the human virus will not grow in animals, so you can't grow it in mice or anything like that. There are rabbit viruses and now mice viruses, but they're not the same as the human papillomaviruses. So in 1992, it was kind of a naive thinking on my part, but I just started to think about this problem as I began my postdoc. And so I asked this question: how do you grow human HPV, human papillomavirus, in tissue culture? And so I went to my mentor at the time, Dr. Lou Layman's, and I talked to him about some of the odd ideas I had. And he said, well, as long as you're not taking too much time, go ahead and try. So there was a technique being used by epithelial biologists at that time where they grew organic typic raft cultures. They called them rafts because initially they floated on top of the media like a raft would float on the water. But now we put them on a wire grid. And this system will grow fully differentiated and stratified epithelium in culture. And so my idea was to try to grow papillomavirus in this system. Well, this had been tried and it didn't work. But I had these ideas, well, maybe if we add these different drugs, like at that time I was looking at protein kinase C activators, and I said, well, maybe if you add those in, they induce viruses like Epstein-Barr virus to replicate. So maybe they'll work on papillomavirus. Maybe that's what's missing out of the system. And we tried several other different things. And in fact, when I did the studies, nobody believed it would work. I didn't believe it would work. My mentor didn't believe it would work. In fact, I got a letter from a longtime mentor, Dr. Robinson would recognize the name, Dr. John Kreider, who told me not to do these studies because he said you're going to waste your career. And you know, a lot of people's careers have crashed and burned trying to grow this virus in culture. But sometimes naivety works in science, you know. You get down with so much dogma and so much things that people say won't work, you're afraid to try new things. And so I tried these things, and actually because we weren't too hopeful, the tissues were frozen away and embedded in paraffin, and we really didn't analyze them for almost 6 months. And a technician came to me one day and said, I'm free this week, and Dr. Lehman says I can do whatever you want me to do. And I said, well, why don't you take those tissues and do some studies to see if, you know, we have anything in them. And that afternoon he came back to me and said, you better come and look at this. This is what drives science. This is where things get really exciting. You know, most of you who are working in labs knows what it's like to sit 8-hour days just filling tubes and spinning things down and refeeding cells. But when these type of things happen, you get very excited. And we had the evidence that we were actually growing the virus in culture. And so this, as Dr. Robinson mentioned, in 1992, we were able to publish this in Science magazine. But now I have virus. For the first time, we don't know how to quantitate it. Nobody's ever had it before, so what do you do to quantitate it? It doesn't grow plaque assays like some viruses. It doesn't grow focal assays like some viruses. So what do? So again, I turned to the herpes virologists who have viruses with the same problem, like Epstein-Barr and Kaposi-sarcoma human virus. And we use their technique. So very simply, using modern PCR, we just take and add a DNase called benzonase to get rid of all exogenous DNA from our viral prep. Then we take and clean up the virus and break it open. So now we have viral genomes, and we just use quantitative PCR and count how many viral genomes. Now this is what you would call a structural titer, but it's the only titer we have right now. Although I do have a student right now working on a biological titer system, and hopefully in a year we'll be able to report that. But now we got to count. We can say we have this much virus. Well, you have virus, you can count it, but is it infectious? How do you measure infectivity? So we take our virus from our rat cultures, we purify it out, and we infect what's called the Hackett cell line. So it's an epithelial cell line. And then using quantitative PCR, we measure a splice transcript from that virus, showing that it's infected and started its replication. And that will give us a measurement of infectivity. So now we're open to do a lot of different things in this way. We can study drugs, we can study some of the life cycle, but we have one little problem. The first culture I ever did had HPV-31. It was a serendipity that HPV-31 was in there. The Rush Presbyterian Medical Center in Chicago was collaborating with us, and they've given us that cell line. They called it CIN-612, CIN for cervical intraepithelial neoplasia, and 612 because it was the 612th biopsy tissue that they tried to make such a cell line from that would work. And so now we can grow HPV-31, but we wanted to grow the more common types and also to do genetics. So we want HPV-16, 18, and even some of the others. So I could get 600, 1,000 biopsy tissues and ask a student to come into my lab and analyze biopsy tissues for 5 years. Or we could figure out another way. Most students, I think, would say, let's figure out another way than doing the cat way. Because with our luck, we keep finding 31 or something. So we asked the question, can I grow other HPV types, and can I do genetics? And so 2 of my former students, Dr. Bromberg-White and Dr. McLaughlin-Drubin, both now have faculty positions at universities, worked on this question with me. So what we did was we simply took our viral genome, we have it in a plasmid vector, we cut away the vector, we use a restriction enzyme, so we cut away the vector, so now we have a linearized genome. We use electroporation, we zap our cells with that DNA, and the DNA goes inside, recircularizes, and then because it's an oncogenic virus, it will induce the primary cells to immortalize. And so we can select the cells that have the viral DNA because they'll keep growing. Primary cells senesce after a few passages, they die out. So if we have the viral DNA in there, they're expressing the oncogenes, which cause the cells to keep growing forever like a cancer. So we select for that, and then we take those cells and put them in our organic typic culture system, and now we can grow virus. And if we have the genome of the type, we can grow it. Plus the fact we're starting with DNA. So if you're starting with DNA with all modern techniques, you can mutate that DNA any way you want. So you can make any type of mutation you want and run the system and see what that mutation does to the viral life cycle, the viral structure, and so forth. So right now, In the lab, we can grow HPV-16. We can grow variants of HPV-16. Not all HPV-16s are equal. There are African variants, European variants, North American variants, Asian American variants, Asian variants, and they're different. Clinically, the African variants and the Asian variants are more aggressive. And so we have several of those. We can grow 18, we can grow 31, and it can go on as long as you have a reason to do it. We've also grown some of the low-risk types, the types that aren't associated with cancer in the system. Plus, now, literally over 100 different mutants have been put into the system and analyzed. And another interesting thing about the organic typic graft culture system is that other epitheliotropic agents can be grown in this system. The herpes viruses grow quite well. We've grown HSV1 and 2, we've grown Kaposi sarcoma virus, we've grown CMV, and now I have somebody growing EBV in this system. Plus, you can grow certain bacteria. Here on this campus, Mycobacterium ulcerans is grown in this culture system. It seems to love the skin, and it'll grow quite well on that. So it is a very efficient system for growing a lot of these different agents if you want to study them in their natural tissue. So, taking a step back, when we grow these cultures, we typically grow them for about 10 days. And this is what a 10-day raft culture looks like, doing a histological stain on it to look for morphology. And we typically, in the beginning, grew them for 10 to 12 days because we knew they fully differentiated in that time and we could get a viral prep out of them. But then I had a student, Dr. Conway, we called him MJ. He's now at Central Michigan University as a faculty member. He asked a very simple question. How can I produce more virus? He was interested in the maturation of the virus and the structure of the virus and doing mutants that would give us some information about how the virus put itself together. And he wasn't getting enough virus to really do the studies in an easy way. And so he asked this very simple question, how can I produce more virus? So he thought, well, we know the mature virus is in the top layers, the cornified layers of the tissue. And we know that if we grow it longer than 10 days, the cornified layer just piles up on top. As you can see here, in 15 and 20 days, you get more and more of that top layer. And so the hope was that that meant there was more virus there. So very simple question, very simple way to look at it. So he grew the rafts for 10 days, 15 days, and 20 days, and then analyzed it. And then from this simple thing, we not only found that the titers got higher up to 20 days, but we also found that there was an interesting maturation process going on in this tissue, that there was a stability change over time, that there was changes in infectivity, changes in the ability of neutralizing antibodies to bind to the particles and neutralize their infection, and changes in the disulfide bonding. And we found that most of this is probably due to the tissue having a natural redox potential, with the lower layers being a reductive environment and the upper layers being an oxidative environment. And so this kind of really changed some of the focus in our lab, and we really started to try to understand this maturation process. And we had really nice demarcations. You know, we could go 10-day, 15, and 20-day, and study what mutations did at those different time points, knowing what the wild type phenotype was. And so that very simple question, how do I get more virus, turned into be a major project that we still do a lot of work on in the laboratory. And has some clinical relevance because the immature virus, even though it's very unstable, is still highly infectious and cannot be neutralized by antibodies, as far as we can tell in the lab. Well, while we were doing all this, others who were trying to find another way around it came up with these recombinant systems to grow particles of HPV. And the first one is the virus-like particles. It was found early on that if you overexpress the capsid genes, and HPV has two, L1 the major capsid gene, and L2 the minor capsid gene, if you just overexpress those in an exogenous system, they will self-assemble into a particle. And so they call those virus-like particles. And this was really important because this is what the vaccine is. When you get the HPV vaccine, all you're getting is virus-like particles. And so that worked really well. And then somebody got the idea, well, what if I overexpress the capsid genes in the presence of a plasmid? Well, we'll self-assemble around these plasmids. And so now you have a particle with a pseudo-genome in it, you can say. And that plasmid could have an expression of a fluorescent marker like GFP or some other dye. And so you kind of have this infection system. You put them on cells and the cells grow green, you know the virus got in. And then somebody had the idea, well, what if I do the same thing, but I throw in a lot of the HPV genomes? Instead of a plasmid, I just throw in all HPV genomes. Well, it'll self-assemble around that HPV genome. So you have what we call a quasi-particle. We have all these different names. I think we're going to run out of them if we come up with anything new. And so there's just been belief that these quasi-particles, and even the pseudoparticles, have the same structure and infectivity pathways that the native or authentic, we've been told to call them multiple different things. Every time we write a paper, somebody wants us to call them something different. But so we kind of stick on native particle right now. So they believe that these things behave the same as native particles. So we just asked a very simple question. We got a little Shakespearean here, and we said, well, when is a virus not a virus? We got all these particles, but what is a virus and what isn't here? Everybody believes these things are viruses. In fact, you'll read their papers. I was always taught here at BYU that a virion was the actual particle, and you couldn't call these other things a virion, but they will call them virions in their papers, and they tend to get away with it. Well, MJ kind of started all this with his work, and then a new student who's actually writing her thesis right now when she's not having morning sickness is Linda Cruz, has been working on this problem quite extensively to try to see if there's differences. And I won't go into them all because there's numerous differences we have found between these particles and the native particles. They are not the same. They behave very differently. But you know, a VLP is able to induce a very strong immune response as a vaccine, so it can make a lot of antibodies, and that will block the native particle. But when you're studying final structure, when you're studying conformation-dependent epitopes, when you're studying infectivity pathways and other things, these other particles do not behave the same way as native particles. And this has been a major contention right now in the field. I've actually told well, at the NIH, they were the big proponents of these artificial particles. And whenever we send a paper for review, usually one of them gets to review it. And so we'll get the other two reviewers, we get a few sentences, and then they'll give us a 5-page review on everything wrong with the paper. And so I've been telling them now with the government shutdown, nobody at the NIH can do any work. So get your papers in now, they can't review them. But anyway, so when is a virus not a virus? Well, I'm going to show you just a little bit here. This is the basic dogma of how a papillomavirus gets into a cell. A virus, as you know, has to bind to the cell, and then it has to go through what is becoming more and more complex, a method of getting inside the cell, so it then get to the nucleus and start making more of itself. So using these pseudoparticles, which we abbreviate PFZ, they have come up with this pathway. So first the virus has to bind to a moiety that has a heparan sulfate on it. Then there's a change in structure, they believe, and the L2, the minor capsid protein, sticks out, and a protease called furin cleaves it. After that cleavage, it can get transferred to this putative entry receptor. Nobody really knows what it is yet. And then it goes inside the cell. So this is their pathway. So we just asked a very simple question. When is a virus not a virus? Well, does the native virus need heparan sulfate? So what I'm showing here, we use different concentrations of heparin. Heparin will block, it'll bind to the heparan sulfate moieties and block the virus from binding. And if you use pseudoparticles, it works really well. But we use different concentrations. And here's our control infection. Not only did it not block HPV-16, but the higher concentrations actually improved blocking. It was almost like it would bind to the heparan sulfate, but it was slowing it down or stopping it from getting in. We also use sodium chlorate, which blocks all production of heparan sulfate on the cell surface. And again, HPV-16 infected quite well, actually better than the control. But then we found out that HPV-18 did the opposite. It was blocked by these heparan sulfate inhibitors. And then we've gone on and tried other types, and they're everything in between, from being totally independent to being totally dependent and something in between. And so the different types, and I think we made a mistake in the field by giving them numbers. No card-carrying herpes virologists would think that HSV1 and Kaposi sarcoma virus are the same virus. But in our field, everybody thinks if you do something with one virus, it works for them all, no matter what they are, whether they're human and mouse. And so 18 and 16, they're only 2 numbers apart. How different can they be? And so we're finding that they're very different. We also thought, well, if it doesn't need heparin sulfate, does it need the furin cleavage? Because remember, it binds to the heparin sulfate, change in structure, L2 gets cleaved by furin. Well, 16 doesn't need heparin sulfate binding, doesn't need furin. And so we did the test. There's a nice furin inhibitor, works very well. We did increasing concentrations of the furin inhibitor, and we found again, not only did it not inhibit infection, it actually increased infection. It was like helping 16 to get in. And, well, somebody said, well, when you're down homogenizing your tissue, the furin's in that tissue and it's cleaving it, so you already got it cleaved. So that's why you don't need furin. Furin inhibitor does nothing because it's already worked. So we put it into our cultures and did it into our homogenization, and we still didn't get anything. So it still did not need furin inhibitor. We then tried a Chinese hamster ovary cell line that lacks the furin gene. Doesn't have it at all. And 16 infects quite well there. We also used the same cell line where they put the furin gene back in. Neither one did anything. And we also even added extra furin inhibitor onto it. Nothing happened. Then the student said, well, we're growing these in foreskin. Maybe if we grow them in cervix, because we can grow the virus in vaginal tissue, cervical tissue, foreskin tissue, tonsil tissue, gingiva tissue, and now we've actually got a GI surgeon having us learn how to do it in anal tissue. So we're covering all the different areas. But if we grow it in cervical tissue, and of course, she has to do everything in pink when it's cervical tissue. The furin inhibitor didn't make any difference. So it doesn't matter where the virus grows in, it still doesn't need it. And then somebody said, well, maybe it's just your lab, because sometimes those things happen. One person's lab can get something to work, the other person's lab can't get it to work. So we actually got pseudoparticles from the NIH, and then we made some of our own. And again, they have that fluorescent protein in there, so when you infect a cell, they glow green, and you can see without furin inhibitor, they're infecting a lot of cells. When we add the furin inhibitor, the infection goes down. So we are doing the experiment right, and we're doing it the same way they do it. It's just that the native particles are different. So this has a real clinical importance because carrageenan is being proposed to be a microbicide for HPV. It would block HPV infections. And carrageenan is from a red seaweed, and it's a heparin sulfate moiety, and most of you probably had carrageenan today in some way, somehow, because it's in toothpastes, it's in creams, it's in ice cream. And this is a great thing because we know it's non-toxic. The human body can take it. So they've been pushing carrageenan, which blocks the pseudoparticles from infecting very, very well. So we did the experiment. Increasing concentrations again of carrageenan. Not only does it not block HPV-16, it actually helps it. And see, we're treating with 10 micrograms per microliter or 100 micrograms per microliter. And we measure the infectivity again with our quantitative PCR assay. But just to point out, the pseudoparticles, their EC50, it's only 5 nanograms per milliliter. We're using a lot more. I think I wrote that wrong. It's 5 nanograms per microliter. That's the typo. So we're using 200 times more, and it still doesn't block. So when you're using these artificial particles for a clinically relevant assay, you have to be careful because they're not necessarily the proper surrogate for that assay. So, now we're going to really stomp on some more dogma here because all HPVs have one and only one protein that's the major capsid protein, L1, only one species of it. And all HPVs, or papillomaviruses in general, have one and only one minor capsid protein species, and that's L2. There's only one of both. They have multiple copies of it, but there's only one species in there of L1. So for a lot of reasons I don't have time to get in, we had some funny data that came up that kept suggesting there might be 2 species of L1 in these native particles. And so another student, who again is trying to hurry to get his thesis done, is Eric Rindock, who took on this problem. And that, I mean, this is like Galileo almost. We were really blasphemous bringing up this information at a meeting that there was more than one L1. Even though if you go back to the 1980s, and of course students think anything over 5 years is out of date, if you go back to the 1980s, there was a lot of 2D gels that were run that showed the evidence that there might be 2 L1s and more than one L2 in a particle. But that was in the 1980s, and we got all these better techniques now, so they didn't know what they were doing. So he took on this question. If you align the N-terminus of the L1 protein with the different HPVs that cause cancer, the first thing I'll point out is this methionine here, which we call the consensus methionine, or number 1, or the third. But this, all those pseudoparticles, all those virus-like particles, quasi-particles, they're made from an L1 protein that starts at that methionine. There's nothing in front of it. They cleaved it all off. So they couldn't make anything extra if they wanted to because they start at that methionine. What we noticed was 18, 16, and 45, just looking at a few, they had a second methionine, 26, that could make about another 26 amino acids on the N-terminus. And then 18 was very interesting. It could add another 35 amino acids on because it has a third methionine upstream. And so there could be a total of 61 extra amino acids on this L1. And we had some funny Western blots that suggested we had a bigger L1 in there, and we weren't sure. So Eric went in and he started mutating these methionines one by one. He did them singly, and he did them in pairs, and then he did all 3. And then we ran them through our system to see what would happen to the particles. So this is just showing infectivity, and you can see here, here's a couple of our controls. Where normal infectivity, if we mutate that way upstream methionine, which we call minus 61, we get less than half of our infectivity. If we mutate the middle methionine, minus 26, it really doesn't change anything, suggesting it's not being used. We mutate out the consensus methionine, we get very high infectivity. We think, but this is a very unstable virus, so we think it gets in and it can uncode easier, so that's why it's more infectious. But we get differences in infectivity, and I can't show you all the data, but the stabilities are different, their ability to be neutralized by antibodies are different, so making these different HPV particles, these mutants, we get different results. Now here's a quasi-particle, which again only has that consensus methionine, and it's just really bizarre. It gives us about half the infection. So he also did, typically let's find the protein. So he worked a long time on these Western blots. And here's our upper band. Here's our bigger piece, our second L1 protein species on the top. But when we mutate that methionine, so it can't make that longer L1, that piece disappears. And we've also used a second antibody to show it. And he's right now in the process of doing mass spec. And both, we can tell you the preliminary data is both of them are L1, and that longer or bigger bigger protein has that extra piece on it. So we have very good evidence there are 2 different L1s at least in the HPV particle. And so again, this is really changing the dogma of the field. Also, I believe we have data that there are more than one L2 in these particles too, but we'll wait till the Fuhrer settles down a little bit and bring that one out. But this changes, how you look at these viruses and how they relate to each other, and maybe second-generation vaccines and treatments can develop from this. So, I don't know, most of you have probably seen these commercials on TV. Merck and GSK, both have come out with a vaccine, although in the U.S. both have been FDA approved. Most people use the Merck vaccine. It covers the 2 benign HPV types, 6 and 11, that are the 90% of cervical condylomas, anal condylomas are due to these types. 16 and 18 are the most common cancer-causing types. They cover about 7% of the known cancers. You can see up here, here's a graph. This is showing 16, about 53, 54% of cancers. 18 adds another 17% on. So again, about 70% of cancers in the world are due to 16 and 18. And then each of the other types, how much more they add on to the cancer percentage. Now, the vaccine works only against those top 2 types, and there's a lot of questions that haven't been answered. The vaccine seems to work pretty good, at least at first. It seems to be giving the protection you would expect. And even though if you go on and Google the vaccine, you'll find a lot of websites that will talk about how it makes women become retarded and all these types of things. This is totally unfounded. This is a very, very safe vaccine. There is a higher percentage of fainting with this vaccine than there are with other vaccines, but we're talking with preteen girls getting vaccinated, so that is probably the reason why we see more fainting than we would with another vaccine. And so that's probably the only difference in this vaccine with others. So it's a very safe vaccine. But there's a lot of questions, like, will it cover everything? You know, it will cover the anal genital types, but will it cover other types of HPV infections, especially oral pharyngeal types? And this is still not absolutely proven yet. It won't work there. The other thing is, there are all these other HPV types up there. This is only protecting against 2. Are you going to get type replacement? Nature abhors a vacuum. You get rid of those 2 types, are the other types going to fill in? Some preliminary papers are actually, even from the CDC, are suggesting the other types are beginning to fill in. That among the vaccinated population, we're seeing more of the non-vaccine types show up. So that could be a problem. Now, Merck is planning to come out with a new vaccine in the next few years that covers 9 types. But that adds a whole new problem set because, you know, with immunology, all that antigen going into somebody, is it all going to work equally? Are you all of a sudden now one of these other types going to become dominant and 16 is going to get weakened, so the major type will come back? So we don't know what's going to happen with that. But a question we asked in the lab, and I took years arguing with Merck on this, that it was an important question, and then finally the FDA told them it was an important question. How long will the vaccine protection last? We're being told to vaccinate young girls at the age of 10. Now, at 20, when they're in college. In fact, in the United States, 80% of women in college will be infected with HPV during their 4 years of college. This is the national average. Not all those infections, in fact, very few will go on to cancer. But a high level of infectivity occurs in college. So you vaccinate a young girl at 10, when she's 20, is she still protected? Now, unlike other vaccines like measles or something, you know when somebody comes down. My mother knew when a person came down with measles. They knew the signs. You get infected, it's an acute infection, the disease comes out in a few days. This is a virus that can go in and infect somebody and be asymptomatic for decades. You cannot monitor the disease and say the vaccine's failing. Because you could say, oh, it's failing at age 40, but it could actually have failed when the person was age 20, and it just sat there latent for all that time period. So you have to have some type of measurement, I argued, that would tell you, what's the measurement in vaccines? You look for neutralizing antibody titers. It's basic virology. I find in the HPV field, a lot of the scientists came from biochemistry or molecular biology backgrounds. Not a lot came from virology or microbiology backgrounds. And so there's certain concepts of infectious disease that aren't the first thing people think about. So this idea of neutralizing antibodies actually took a lot of them by surprise. We're going to do what? So we have the particles, so we can do this assay. We can take our virus, mix it with serum from women, and see if it's neutralizable. And so Merck actually agreed to an investigator-initiated clinical trial where they gave us some serum samples. And so we were asking, how long will the vaccine protect? And so my technician, Janice Malisi, has been working on this project. So we got serum from the Merck. They gave us serum from these women in each of these graphs. Well, on the one side, see how I got this? Yeah, on the one side it's one woman. We're measuring against 16 and 18. Top is 16, bottom is 18, and on the other side is the second woman. But we have 60 of these samples. So they gave us pre-immune, so before the vaccine, that's what this first said. We diluted the serum 1 to 200, 1 to 2,000, 1 to 20,000, and looked at how protective it was. So anything below the line means that it blocked infectivity. Anything above the line, for simplicity's sake, says that the neutralizing antibodies are still strong. I mean, the neutralizing antibodies have weakened, they've gone down in numbers, and the vaccine's not protective anymore. We also got serum from the same woman at 7 months post-vaccination, at 4 years post-vaccination, and at 9 years post-vaccination. So we tested the same way, 1 to 200, 1 to 2,000, 1 to 20,000 dilution. We also had our negative control, no serum, no antibodies, so that's how setting, just the virus, showing how infectious it is. And then our positive control, where we added a confirmation-dependent neutralizing antibody that would just completely knock out infectivity to make sure everything was working properly. So you can see from this that over time, it starts off working very well, but over time, we start to lose the ability to neutralize. So one thing I found out from Merck is that you can't say booster anymore. It's a dirty word to the vaccine companies. I don't know why, but it is. But you can say challenge or fourth dose. And so we're going to propose to them when we get this written up that they do need to have a fourth dose. And we need to do some more studies on this. So the titers are going down. And like a lot of vaccines, well, you could argue those who are immunologists out there are thinking, well, but you got memory cells. The antibodies may not be out there, but the memory cells still remember what everything's about. So when you get reinfected, the memory cells kick in and they make more antibodies and they protect you. But most viral diseases start at a site that's not the target site for the disease. You get infected by measles, you get infected for something else. It starts at one site and you get some symptoms, maybe a fever or cold-like symptoms. The virus goes viremic, and then the memory cells kick in and block it from getting to the target tissues where the real disease occurs. This is a virus that goes right to the site where it wants to be and stops there. Doesn't need to spread, doesn't need to have that second spread. And so the question is, will memory cells do anything? Because once it's inside the cells, the antibodies aren't going to do anything. And so this is a really important question because we're going to have a population of young women and even young men, because it's been approved for men, who are going to think they're protected, and it's not. It could be that it doesn't work for them. So this is the last question I'm going to deal with today. Well, I have one more most important question at the end. But this question is, what disinfectants will kill HPV? I never thought I would get into the clinical or translational work. I thought I'd always be basic. So, but now that we have virus and we have all these assays, we can do the tests like neutralizing antibodies and look for drugs and microbicides and stuff like this. So Dr. Robinson here, one time I was out here, he said, well, he works with a lot of disinfectants. And he said, yeah, nobody's ever looked at HPV. They can't put it on their label because nobody knows if their disinfectant works or not. You can do the test. We can work with you. We could do this and study this. So him and one of his students, and I'm just going to point out, some of you might remember him. Out of my 4 sons, only one stayed in this field. The rest went running and screaming as far away from this as they could. And maybe we dropped him too many times, I don't know. He's here, and he worked with Dr. Robinson on this project. And so we asked that question, what disinfectants will kill HPV? Because in any hospital, in any dental clinic, they don't know. They're using what kills other viruses or what somebody thinks will work best. And so Jordan started this question. He did all the studies here first. And because it's such an important question, we did all the studies again in my lab across the country with different people, different stocks of virus, different cell lines, and everything else, because there are thousands and thousands of lawsuits out there pending where women are suing their OBGYN saying, I got the virus from you, from your unsterile equipment or something like that. And how many dental clinics are passing this virus around? So we did the study. We had to adapt our infectivity assay. So we would add in our disinfectant, and then we'd have to add a neutralizer because a disinfectant's going to kill the cell line if we leave it on, and filter our virus to get rid of it all. Then we do our infectivity and measure by quantitative PCR again how much infectivity we have left. So we got a really surprising result. Anything above the red line means it's blocking infectivity. So anything at 1, that's a 10-fold reduction, 100-fold reduction, 1,000, 10,000, 100,000. So the first thing you see is there's a lot of these disinfectants that aren't working. Second thing is we use the quasi-particles to compare, these artificial particles, and we see that the native particles and the quasi-particles, don't always match each other. The causing particles tend to be more sensitive to some of these disinfectants like isopropanol and phenol than the native particles. So you see all this blue below the line, that means the native particles. These disinfectants, if they're doing anything, they're just giving them a nitric bath and they're, doing better. We did find a couple, this peracetic acid at 1.2% and bleach, which was a nice thing to know, actually will kill both of them. And they're both oxidizing agents, and so that's how they're working. Our lab switched over. We were using one of these other solutions to clean everything in the lab. As soon as we got this data, we started using bleach on everything. But the more important thing here is these 2-glutaraldehyde. This is what all healthcare facilities use to clean, to kill papillomavirus. All the OBGYN offices, all the dental clinics, everybody, if they're going to use a disinfectant, this is what they use because it believes. When I showed some of this data in my medical school class, I had the safety officer had sent me an email before I got back to my room because it got back to him that I was saying this. And he showed me all these papers, and I said, have you read any of those papers? And I pointed out to him that every single paper said, we think, but we don't know, that these things are killing. So actually in our medical center and all the satellite clinics, they've switched over to disposable equipment in the OBGYN clinics. We're not saying that they did get it from their doctor, but all these women are suing, it's a possibility. So there's a possibility of a nosocomial or a fomite infection, that objects can carry this virus around and they can reinfect other people. In fact, we know that people with genital infections, if you take and swab their fingers, at any one time, 20% of them will have the papillomavirus on their fingertips. And those Purell containers that everybody has to squirt on their hands with all the different scents do nothing to kill this virus. And I've been told we should study hand washing. We got to figure out how to do that with soap and see what it does. So this one question here, okay, I got to go quick. We noticed that a common virus called adeno-associated virus type 2 was killing papillomavirus infected cells or cervical cancer cells. So we published that. It was inducing apoptosis or cell death. So we asked, will it kill other types like breast cancer? And so we used the highly aggressive breast cancer cell line, and we showed that if we infect those cells with AAV2, those cells die. And we've actually done the mouse system now where we form tumors in the mice with these breast cancer cell lines. And if we inject AAV2 into that tumor, it becomes very necrotic. And we've actually analyzed the molecular pathways. It's doing the same thing that we see in the Petri tissue culture dishes. And so they become very necrotic. They get this massive eschar scar on. But now, the most important question. All of you out there, everybody works on some infectious agent, they think they're number one. We actually have proof that we're number one in HPV. First off, we were one of the first viruses ever put on the government list as a known carcinogen. HPV and hepatitis B and C were the first on that list as a government-approved carcinogen, I guess. Harold Zerhausen won the Nobel Prize for linking HPV to cervical cancer. Now, I know what you're saying. He won the Nobel Prize. Why don't we have him speaking? Why do we have Dr. Meyers? I have something that makes him jealous. I graduated from Hogwarts, for one. Also, if you look at the Uncle John's triumphant 20th anniversary bathroom reader, and I want to point out that this is not just your ordinary bathroom reader, but this is the 20th anniversary triumphant bathroom reader. Then you go to this page, you will find Penn State College of Medicine Professor Craig Meyers is quoted in that book. So you have to be careful what you say because you never know where it's going to show up. I know this makes Dr. Zerhausen very jealous every time he hears about it. We also have our own castle in Germany. No other infectious agent has their own castle in Germany. Here's Wartburg in Germany. Finally, because you're scientists, actual numerical data. Popular science asks, what is the worst job in science? An anal wart researcher was number one. So, we have absolute proof. What's really scary is nurses are on this list too. These are all the different people who work for me. I didn't get to talk about all their different work. Dr. Christensen at Penn State, who's been my longtime collaborator. Also, all the funding sources and collaborators. For my AV2 work, I get donations. I've actually collected over $200,000 in donations from companies and organizations, schools, and individuals. And some have donated up, actually had pancake breakfasts and stuff, and donated up to $5,000 or more dollars. So I never thought that would happen. So I want to thank all my people who have contributed to the work, and thank you for your attention and for this unbelievable honor that you're paying me today.